Hello, everyone. My name is Wayne, and I'm a PhD candidate at the Divine Lab here in the Center for Brain Research. Today, I'll be presenting a poster on an interesting way to study post-war platelet functions in trauma transfusion using an in vitro transfusion model. As many of us are aware, the current model of storing platelets at room temperature leads to a very short shelf life of just 7 days. The units are also prone to bacterial growth over the storage period, given that it's storing at room temperature. Furthermore, while the room temperature stored platelets are optimal for patients requiring prophylactic transfusions, these room temperature stored platelets aren't necessarily the best for treating actively bleeding patients, such as patients in trauma. There have been renewed interest in bringing back cold store platelets due to their superior hemostatic function in vitro compared to room temperature stored platelets. And since these platelets are stored at lower temperature, they have the potential to slow down bacterial growth and platelet metabolism to achieve prolonged shelf life of platelets. So these benefits of cold store platelets also make them very attractive for treating actively bleeding patients. So in this study, we designed an in vitro transfusion model to specifically compare the difference between using room temperature stored platelets versus using cold store platelets in a massive transfusion protocol to treat bleeding patients. So in our model, looking at the methods, we combine equal volumes of thawed AB positive plasma with stored red blood cells and either room temperature stored platelets or cold store platelets. So stored platelets were sampled on day 1, 7, or 14 to make transfusion packages. Transfusion packages made from room temperature stored platelets are labeled as warm packages, and transfusion packages made from cold store platelets are labeled cold packages. Now, to simulate what happens to a trauma patient's blood during massive bleeding episodes, we simulated one of these conditions by first obtaining whole blood from volunteer donors, and then dilute the blood to 20% hematocrit using physiological saline. So this is our hemodiluted condition. We also simulated hyperfibrinolytic whole blood, which also happens during trauma, by adding recombinant tissue plasminogen activator to the volunteers' donors' whole blood. So this is our hyperfibrinolytic whole blood. Then to conduct the in vitro transfusion, we mix 70% of our transfusion package, either the warm or the cold package, with our modified whole blood. And this mixture of whole blood is analyzed on rotational thermoelastometry machines to look at their clotting characteristics. So moving on to the results, here you can see that these black bars. So these black bars are the modified whole blood ran without any addition of transfusion package. So the first thing we notice for our results is that looking at the hemodilution results is that both warm and cold package transfusions significantly improve the row time parameter of hemodiluted whole blood. So this is in comparison to the whole blood that didn't have any transfusion package addition. However, we did notice that the whole blood with the cold package transfusion resulted in longer clot formation time and less maximum clot firmness. So this is quite contrary to what we see in the literature. However, the one possible explanation for this is that since we're adding 1:1:1 volume ratios, and the cold store platelets have a significant decrease in count over storage period, this difference that we see here is likely due to the different platelet concentration between our warm and cold packages. So moving on to the results of the hyperfibrinolysis whole blood. Very similar to what we observed in hemodilution results, we see both warm and cold transfusion packages significantly improve the row time parameters of our hyperfibrinolytic whole blood. So in particular, we see that only the cold packages transfusions, but not the warm cold package transfusions, led to a significant decrease in the maximum lysis of our hyperfibrinolytic whole blood. So in conclusion, cold store platelets stored for an extended period of time may be used to treat actively bleeding patients. They might also provide additional benefits of resisting hyperfibrinolysis that is common in trauma and massive bleeding. Thank you.